Alright, to begin, we'll start with the transcription process. During the transcription process, we start with the general transcription factor, which we put onto the title box. We then start with the mRNA polymerase, and we attach it onto the DNA. Soon after we finish with that, we move on to the process of initiation, where the RNA polymerase makes a copy of the lower DNA into a single strand of RNA. After the process of initiation, we move on to the process of elongation, where the RNA polymerase continues to copy down the the DNA into RNA all the way to 3'. Soon after the process of elongation, the RNA polymerase then removes the RNA strand out, allowing us to start the process of RNA processing. During the RNA processing, we start with 5' capping enzyme, which adds guanine to the 5' end of the RNA. And similarly, we have the poly-A polymerase, which adds adenine at the end of the 3' end of the RNA, giving us a poly-A tail. Soon after that, we get to the process of splicing. During the process of splicing, we have a protein complex enzyme called spliceosome, which cuts off the introns, like so, which allows the pre-RNA exon to rejoin, completing the mRNA. The complete mRNA is then ready to move out of the nucleus and into the cytoplasm for translation. After the process of transcription, we start the process of translation. And the purpose of translation is to decode the mRNA into the ribosome structure, which then creates a polypeptide bond. And this polypeptide bond is later formed into an active protein, and it does its function in the cell. Now, to begin, we start the process of initiation. So, the mRNA is inserted into the ribosome, where it starts in the P site. The P site is where the peptide bond site is located, and this is the A site, which is the amino acid arrival site. After we're done with the process of initiation, we start with the process of elongation. The process of elongation is that we start with our start codon, which is AUG, and we find its tRNA and its amino acid. We put them together, and we attach it onto the start codon. So right here. Soon after that, we then find our next tRNA and our next amino acids for our A site. In this case, our tRNA will be UAG, and our amino acid will be this. Soon after that, what's going to happen next is that it's going to continue to move through, exiting through here, which is the E site. So we push this down onto the next codon, empty codon, where soon the tRNA leaves, leaving this in the P site, while the amino acids are overlapping. Once they're overlapping, this is where the polypeptide bond starts to form. And we continue to do this process. Until we get to the end of the codons. So We continue this process all the way down till we get to the stop codon, which is UAG. Once our UAG, which is considered as a stop codon, meaning that the whole process stops, and all the amino acids that's overlapping are now turned into a polypeptide bond. Okay, come on.